Good morning, everyone, and welcome to today's webinar, The Emergence of AI-Guided Genomics to Accelerate Variant Interpretation. My name is Garrett Sheets, and today we're joined by two members of Genomenon's leadership team who will guide us through a discussion about where artificial intelligence meets precision medicine. We'll unpack emerging innovation around AI-driven genomics and how it's improving outcomes in both the research and the clinical space. We'll also discuss key findings from a recent study on the Mastermind Genomic Search Engine and talk about how using a computationally intelligent approach to curate genomic data has quickly surpassed almost 2 decades of manual effort. They have a lot of great information to share, so I'll get right to housekeeping. For those watching, feel free to put your questions into the Q&A, and if we have time, we'll get to those at the end of the presentation. This webinar is being recorded, and we will email you the recording within the next day or so. So without further ado, I'll briefly introduce our speakers, and they'll tell you a little bit more about themselves. We have Dr. Mark Keel, who is our co-founder and chief science officer, and we also have Dr. Brittany Jones, our director of customer success. Hi, Mark. Hi, Brittany. Thank you both for joining us today.
Yeah.
Brittany, I'm gonna hit the ball over to you. Why don't you get us started?
Yeah, great. Thank you very much, Garrett. So my name is Brittany Jones. I'm the director of customer success. Most recently, I was at Fabric Genomics. So I just joined the Genomenon team. Most recently at Fabric, which is a tertiary analysis software. So really looking at a clinical support software, but also in the world of AI, which is why I'm here with us today. My history is that I've spent the better part of over 10 years in the genetics and genomics space as an application scientist. Before that, I got my PhD at UCSF. Mark?
Yeah, thanks, Brittany, and welcome to Brittany. This is one of the fastest hires to webinar cycle that I've ever seen. So thank you, Brittany, for joining me. I'm Mark. I'm the Chief Science Officer and founder of Genomenon, and my background is as an MD, PhD scientist, having done clinical genomics, both cancer and constitutional, as well as doing a fair bit of research genomics. And I'm a data nerd. I love data visualization, compelling order to chaos, and hopefully that'll come out in the conversation that Brittany and I are going to have. But I'm the Chief Science Officer here at Genomenon, and in that role, I manage teams that supervise the data quality that we produce, as well as the content curation that I'm sure will come out as Brittany and I go through some of our questions here. So welcome to all of our attendees. It's a pleasure to have you. We'll welcome questions as you have them toward the end, but otherwise, Brittany, let's take it away.
Yeah, absolutely. Yes, we wanted to get kicked off today just with a little bit of, can you give us a background on Genomenon and maybe how, since this is about AI, how AI figures into the platform?
Yeah, sure. So apologies to those of you who know Genomenon if this is a little bit redundant, but we have a much wider audience here than is typical of our webinars. So let me tell you about Genomenon a bit, Mastermind, our product, how we deliver value for variant interpretation, both in the clinic and in pharma, and then how AI contributes to the value that we generate. So at Genomenon, we are organizing the world's genomic information, and we're doing that in the service of saving and improving lives of individuals with genetic and other related diseases. And so I suggested that Mastermind is our software, but it's also our database, and that's where the value really starts. It is a database of evidence that we have indexed from the published empirical medical literature, the clinical and research literature. It comprises titles, abstracts, full text articles, including figures and tables, as well as supplemental material. And that's updated on a weekly basis and goes back in time many decades. And it's a highly sensitive data repository. But one of the measures of the sensitivity is a very sophisticated algorithm that we use to pull out information from those references, to organize it, to annotate it, and then to present it to our users. And so we sort of cheekily referred to that as genomic language processing, which is a slant on natural language processing, which we'll talk on a little bit later. And we like to say that there's nothing natural about genomic language. And so the genes and the way they're described, the variants and the way that they're mentioned, the obviously multiple ways that you can describe a disease or clinical phenotypes, the drugs, etc. all of that is part of our genomic language processing, and we organize that data into genomic associations for our software users to see. Increasingly for our higher throughput labs to access that data in higher throughput through an API and custom configure how those data components get put together. And then recently, as of the past, say, 3 or 4 years, we've actually begun curating that content as well. So using a sort of unique combinatorial approach of automating the indexing of that information with our GLP and computational capability, and then expertly curating that information to ensure the utmost accuracy of that information, as well as the specificity and relevance to specific questions or different diseases or therapies. We do that through a manual curation. And then to answer the latter part of your question specifically, Brittany, how do we use AI? So AI is a loaded term. It's very broad. I've likened it before to saying we're doing science. It's just kind of contentless, and it's taken on a bit of a life of its own. But our general approach to using AI, our philosophy is that we don't use it at runtime. We use it while we're learning. So on the back end with our very capable development team and our content and scientific team, we use AI capabilities to understand the patterns in the data and then to take action on it. So nothing that you see in Mastermind is sort of live AI, but we're leveraging AI in the background very, very frequently and very sophisticatedly to understand these patterns to most effectively prioritize the content, both for our software users as well as for our internal teams. So
yeah, and that kind of brings up another question there. Like you just said, AI is everything. AI is science, right? You know, what is the definition that you're giving of AI? Maybe a little bit more around like, you know, machine learning applications.
It's a good question. So there's often a confusion about AI and machine learning being the same thing, and it's actually machine learning is a square to the shape of AI. So machine learning is a very, very narrow. It's broadly used and very powerful, but it's a narrow application of AI. So this is not meant to be detailed technical conversations because we want to put it more to the practical applications in clinical genomics. But I like to sort of fractionate AI into things like interactive AI, an example being, you know, chatbots, for better or worse. And then functional AI, the things like Boston Dynamics and the awesome dancing robots that are also highly useful, but it's really cool and somewhat intimidating to see them dance. And then things like, you know, visual AI, you probably have heard about deep fakes and, you know, augmented reality, etc. But the one that I want to focus on, just to give you a landscape view, is analytic AI. And that has to do with finding patterns in data. And a subset of that subset of AI is machine learning. But we use machine learning at Genomenon, but not as much as we use something called named entity recognition, which again is a facet of natural language processing, and specific applications in genetics and genomics would invoke the GLP that I talked about before. But just to put a finer point on it, for analytical AI, I like to say it's using statistics and data to find patterns to ensure that the evidence that you're organizing and the answers that you're coming to are appropriate for a user question.
Yeah, and so that the other question that your original statements were bringing up is you were saying that we're not using AI when we're using it as part of the learning, not as part of the final product. So that kind of brings up what are some of the challenges of bringing AI into clinical space, right? Into a clinical practice, and how do we push that more? Like, how can we get the people to understand that, feel comfortable with it, and then bring that in-house to use the power of AI?
Yeah, so there's a huge difference between the acuity of the result, the importance of getting it right on a clinical side and on the consumer side. And so I'm an avid YouTube watcher. I watch lectures as I fall asleep. I listen to music while I work. And there's uncanny algorithmic predictions that come up from YouTube as they track my search data and that sort of thing. And that's AI. But if they get a video wrong, I just skip over it. If I've seen it before, they might know that I like to rewatch stuff. It's very low stakes there. Similarly for, say, Facebook ads. If you do a Google search and then you see a Facebook ad. In the clinic, it's totally different. The data has to be right. And so when I talk about not applying AI at runtime, it's because we want to ensure that there's reproducibility and predictability in the results that are generated. And that's a challenge with typical applications of AI. There's actually benefit on the consumer side to having some of that serendipity, and it's a huge burden and a problem on the clinical side. So there's that, is you've got to get it right. There's also the need to access the evidence, and there's a trend in AI for these to no longer be black box tools, but actually defend what they've said. So you can imagine if the YouTube algorithm had to tell you why it gave you that video and show you all of the machinations that it undertook from analyzing your search data, that's not wanted or welcome and a problem on the consumer side. That's exactly what you need to do on the clinical side. We as clinicians who are looking at this data need to know why it's being prioritized. And then the last thing I'll say fairly briefly is related to showing the work, which is something that Genomenon puts a lot of primacy on, is there's got to be more of a interconnected dynamic interaction between the end user and that evidence. And again, that's what we make available to our users through the software.
Yeah, and so I think that was an important point you just brought up, which is that user input, right? So they can actually, that last piece, they can actually put in more information. And I know you had a couple of examples of those that I'd seen where the user searches with genes associated with leukemia.
Yeah. So this is one of my favorite examples. It's harkening back to my training in my research, and much of my clinical practice was around hematopoietic malignancies. So studying them and diagnosing them. And obviously Mastermind knows the genomic associations around leukemia, say. We'll just keep it generic. Well, so an example search might be going into a tool like a database tool, a genomic association capability like Mastermind, saying, I want to see genes associated with leukemia. So that's a pretty open statement. And as a user, you might know exactly what that means to you, but unless and until you let the algorithm know what specifically you care about, it's showing you everything. And especially in the context of clinical result returns, it's being very sensitive on purpose because it doesn't want, I'm sorry to anthropomorphize the algorithm, but we don't want the user to be missing out on anything. So an example of a couple of results that might be returned are the ones that probably people are expecting would be like c-KIT, FLT3, CBP alpha. Those are all mutated to cause cancer, but there's also structural variants through gene. Those are gene associations with leukemia, like BCR-ABL, MLL deletions. There's also surprise entities where, say, CD34, which is used as a marker to identify stem cells for therapeutic transplant. Well, that's a gene too. And so that gets at the heart of what you said about the user obtaining results that are predicated on the quality of their input, how thoughtfully they've told the algorithm what specifically they care about. That comes out in the results that you have, but it has to do with that competition between sensitivity and specificity.
Yeah, which brings us forward exactly to what we wanted to discuss next, which is technical challenges. So can you speak to some of the technical challenges, especially in genomics, right? From that data perspective, I mean, there's just so much data, right? So how do we make those types of associations, and then how do we drill down?
So there's two flavors of that, actually, the proliferation of the data, and they feed off each other, right? The one is there's this hyperproliferation of data in the clinic and in the research world. Just sequencing has been commoditized. Now the biologist isn't constrained and they can say, I want to sequence 1,000 patients with this disease. It's not a monetary or technical barrier. It's just sort of an organizational challenge to find these patients and have that happen. So there's this dramatic expansion in that data. And there's the patterning of that information then in the medical literature. So it's organizing all that data as it's growing in the clinical lab, and then reconciling it with that proliferation of the evidence that's been published. And we'll talk about some more of those challenges here in a second, I think, but there's that just creates a situation where there's a need to automate. And automation is the real challenge, where before, I've said before, when I trained, it was you'd look at a single mutation result on a paper requisition. And you can't do that with a panel or an exome or a genome. There's just this critical need to marry the research data with the sequencing data highly computationally, and it has to be automated. And then all of these disparate aspects of the genomic pipeline need to be seamlessly integrated. There's a lot of moving parts. I don't know if I imagine that you coded a little bit and took the raw data and put it through its paces and processed it. I mean, it's not as easy as it looks at the clinical user level, where you just click a button and out comes the data. So there's challenges to streamline that integration. And then as I was training as a clinical pathologist, one of the most interesting teaching points from my mentors was the critical need for reproducibility of the data, reproducibility of the output. It has to work predictably on a Tuesday morning and a Thursday afternoon, whether Bill or Sue are manning the shop. And so that reproducibility is a real challenge when you're automating and dealing with all this data, especially if it's so proliferative and disparate. It's different types of data. So that's a flavor of some of the technical challenges.
I mean, yeah, as part of your certification process, they often make you prove that, right? Like in these clinical labs, you have to prove that if Joe did it one day and Sue did it the next, that we're getting the same output, so that patient 1 isn't treated differently than patient 2 just by accident.
Yeah, and AI, there's not much room for the typical voodoo that is AI, where a little bit of overfitting can totally change the result.
Right.
And so, as I said, to put a fine point on this in the context of the conversation, there has to be much more of a management of a curation of the application of the AI in the context of clinical genomics.
Yeah. And that's sort of what you were talking about in how do we make sense of all of this data and how do we organize it. So, I mean, people have been trying that for a long time, right? There are other databases out there. So to dig into a little bit more, like, can you speak about some of the challenges of organizing that data? I mean, you've got your ClinVar, your LSDB, your Cosmic, there's a lot of other databases that have been trying to do this to provide this back to interpreters.
It harkens back to this superabundance of information, but also the complexity of that information. So we talked enough, I think, about how proliferative this data is. Let's talk a little bit more about the complexity. It's I remember first as a graduate student, first beginning to use DBSNP, and how it was it's sort of an embarrassment of data. There's just tons of data there. And it's now, when I was a graduate student, and I think still, it was being used for things that it wasn't intended for, because there was so much data there. But there were sort of, there were things that were being inferred about the data that weren't exactly accurate because of the nature of what was submitted. A lot of research samples, a lot of engineered variants. I mean, we've clearly grown up since then, and now we have more appropriate databases of population frequency, et cetera, that address those issues. But the challenge is, with this proliferation of data, who's going to do the work? And there's typically a distributed model, which is what happens with ClinVar, where the organizers of ClinVar have tasked the community with submitting and qualifying and interpreting those variants. And it's a repository of that information. They've taken some pains to ensure the quality and annotation of that data, but it's an ongoing challenge. And so that's a general database of genetic variants, but then you talk about locus-specific databases, which, if you're not familiar, is a disease or a gene. And typically this happens out of an academic setting where one researcher takes it upon themselves to pick up that mantle and organize that data. Well, it's really hard. It's very complicated. If you have to keep track of the literature as it's published, go in and disambiguate the variants that these authors have described and typoed or described it in a slightly different way that makes it hard for you to reconcile with already existing entries in your database. And so the challenge is, I think, the complexity and the need to have more high throughput, but also the need to have it be manicured and expertly curated. So those are the two sort of competing challenges, I think, with organizing these variant databases.
Yeah, and so I know that in the paper it actually mentioned, so we'll kind of go into the paper now a little bit more, but in the paper it actually mentioned that, Genomenon's finding 5 times more, or about 5 times more pathogenic variants than, say, ClinVar. What do you think is leading to that challenge, or what do you think is leading to that discrepancy? I think it was 10 times more variants overall, even.
Yep. Yeah, so let's talk about that a little bit more. So a variant is not a variant is not a variant. I mean, right. you can have a variant that's a polymorphism and is benign and doesn't contribute to disease, and you can have a very well-known, pathogenic variant that is widely studied and everybody who is in biology knows that it's associated with disease. But there's this wide distribution in between. Mm-hmm. And so there's the need to be as comprehensive as possible, because especially with that proliferation of evidence, any one study might contain, controversial evidence, properly evidence that changes your view about a variant, or provides information for a rare variant from a rare case, or a rare disease circumstance. And so there's that wide distribution and heterogeneity of the variants. But when you talk about how Mastermind, our database of this evidence, compares with ClinVar, the reason that we're able to surface more evidence and find more variants is because of our computational approach, which is you sort of put them next to each other. Our model is computational, and the ClinVar model is crowdsourced. And there's challenges with ClinVar in the incentivization of having labs submit and users submit. But even if that is not a challenge, there's this hope that you would have all of the patients that have ever been seen, which we've amortized across the decades of medical literature, captured in that database. And effectively, because ClinVar is relying on users submitting evidence from real patients, there's a lag, there's a substantial lag. And so, as you point out, there's maybe 10 or 20% of variants that are captured in ClinVar. And there's a bias toward well-known variants or polymorphic variants on either side of that spectrum, whereas our reach into the literature and our computational capability captures everything, including that hump in the middle, where many of them are pathogenic, as you point out, but a substantial number of them have been found in the context of patients. There is evidence that they might be associated with disease. And any, some other paper, functional study, a new cohort or whatever, will tip that balance, and the evidence will be sufficient to say, yeah, this is diagnosable.
Yeah, and so you keep saying Genomenon's computational approach. So, I mean, what is Genomenon's computational approach, and what does AI have to do with that?
Yeah, so good question. I've alluded to that a little bit before about our data substrate is the empirical literature. This is now talking about the Mastermind software. So the data in the software right now is predicated on those publications. GLP is very sophisticated in knowing it's bioinformatically literate and aware. It knows about legacy nomenclature and different transcripts. It knows about HGVS and how authors don't often respect HGVS nomenclature, which is the standardized way to describe variants. We can take a step up too and talk about structural variants, and copy number variants are particularly challenging to search for, to find in the evidence, but also to be sure that you're not missing something that's similar but not identical to your search for variant. And so that's what GLP does. And recalling that I described named entity recognition as a facet of AI, GLP is totally focused on named entity recognition and expert at organizing that information. And then I alluded to one of the ways that we deliver value to our clients through curation of this content. Mm-hmm. And your question here allows me to say that our ambition is to curate the entire genome. Mm-hmm. So every gene in the context of every disease, every variant, every scrap of evidence that's been published about those variants. We have ambitions in the coming years here to curate all that content. We've begun with a targeted approach for some of our pharma clients, and we've also taken a disease, a body system by body system approach, beginning with neurodegenerative disease. So we've curated all the genes. There's 36 or so genes associated with ALS, neurodegenerative disease, Parkinson's. There's a couple dozen, Alzheimer's, et cetera, et cetera. So we've used our AI approach to find all the variants and all the genes that are associated with those diseases, to organize and annotate that evidence. And then there's even sophisticated computational capability that would fall under the umbrella of AI that allows my team to curate that very, very quickly and highly accurately. And so that's, again, without going into much technical detail, that's a flavor for how we're using AI to mitigate these challenges that we talked about with the manual or distributed but still manual approach to organizing data, like in ClinVar locus-specific databases.
Yeah, and I think up until now we've been focusing a lot of the conversation around, well, ClinVar. I mean, I know it has both somatic as well as constitutional information, but I know this probably is even more expounded in the cancer space, right? So when we're looking at so there's heterogeneity and there's varying mechanisms of disease in the constitutional space or the hereditary space, but
yeah.
Are there similar challenges? Are they different? Are they bigger in the somatic space?
Yeah, so you hit the nail on the head with respect to heterogeneity. Every constitutional disease, by and large, has its own idiosyncrasies and weird way that it causes disease, with a focus on genes. And obviously, cancer is not simple, but biology is very economical, and it is unabashedly reusing mechanisms to cause disease across multiple different disease types. And so the challenges that occasions is that an exaggeration of the proliferation of that data. So in typical searches of cancer-related data in Mastermind, say, there's a lot of results. And the questions that the user is asking now are less about, is this a variant that causes disease, which is typically the case in constitutional. It's now more about, what do I do with it? And what specifically do I want to ask? And it would be things like, well, what diseases has it been found in? Is there a prognostic significance? What do I treat this patient with? What's the latest guidelines? Is there any untoward side effects, counterindications in this context? And so it's a greater depth of evidence and a somewhat broader need to get more specific information and evidence out. Those are the sorts of things that I see as being different challenges in the cancer space. And for our AI capability, I mean, sensitivity is less important. It's still important, obviously, and especially for the rare variants, but it's less important for like a BRAF V600E variant. Everybody knows that it's causative. It's now about prioritizing the information, just like the leukemia example I gave. Well, what do you want to see? So giving users capabilities to refine their search in a way that hyper focuses their attention on the most meaningful evidence to make their downstream clinical decisions. So that would be what I say is the pivot, is more of an emphasis on specificity because of that greater depth of evidence.
Yeah, and you just mentioned this. So you gave two different scenarios of two different types of variants, right? So we have these ultra rare variants, right? And then we have variants that have been published 18,000 times, right? And so there's two challenges that are going to be associated with these two situations, right? So as a variant scientist, I am not, but let's pretend for a second, I'm a variant scientist, right? So I'm going in there and for the rare variant, I want to make sure that I find those two papers that mention that variant that could be associated with some type of phenotypes, diseases, whatever you want to say there. So there's that challenge as opposed to the challenge of there's 7,000 references that I need to get to, and I need to actually figure out which ones are important and what matters, right? So can you describe how AI can be used in those two opposing, I won't call them opposing, but those two scenarios?
Yeah, it's a great question, and just recalling the information that was recently published by my team and I, I think, and this is corroborated by some of our user input, when they've taken Mastermind to task and asked these questions of, archived variants or even real-time variants and using their old method of searching, did they find everything that they needed for every patient with any one of these variants? And the standard yield for that rare variant that you said, you talk about a needle in a haystack, is, am I absent an AI capability like Mastermind? Am I missing evidence? Well, that you are. That happens 10 or 20% of the time. I think the paper cites a statistic like 14%. Mm-hmm. But in our experience talking to users, it's not small. It's 10 to 20% of cases where you do find evidence, and it is instructive, and it only comes out of the results from this very sensitive capability in Mastermind. So that's on the first side, and that's where we talked about the need to ensure that you're maximally sensitive. But then on the other side, where you talk about, well, suppose it's a widely published variant. Well, I mean, at the one extreme, it would be V600E or similarly popular variant, and there it goes back to what I answered before about, well, what do you want to find? Mm-hmm. But a more typical case, and I think the search results that are most typical in Mastermind are between one paper and a dozen, so about five papers. Well, if you'll permit me to describe a little bit more about the standard guidelines mm-hmm. to interpret these variants, on mm-hmm. the constitutional side, it's ACMG, mm-hmm. which requires population data and predictive data, and then publication mm-hmm. data. Now, AMP is a little bit more focused on the guidelines, but it still benefits greatly from finding these functional studies or other related clinical studies. And so for much of that, the crux of evidence needs to come from the published literature. And the ACMG guidelines in particular, if you have one study that's well performed and appropriate, but one study can tip the scales away from a variant of uncertain significance to a likely pathogenic variant. So in the context that you talked about, where, it's been published and it's in ClinVar and there's a paper or two, it actually becomes really important that you're not missing the other two papers that describe it, mm-hmm. because those other two papers, if they're obscure, if they're in a foreign language, or if they're published, mm-hmm. a decade ago or whatever, those can wind up being critically important. And so that number of 10 to 20% actually is just the tip of the iceberg for the importance of sensitivity, which can only come, as I've alluded to, with a computational or AI approach to organizing this data.
Yeah. And I think the next thing that it actually enables, so we went through, again, me as this variant scientist, I went through all of this information, right, that first time, and I classified this variant. Well, often in the first scenario that we just talked about, there wasn't enough evidence. There's that one paper, but it doesn't really mention how it's affecting in the disease, or we're not really sure what that variant's doing, right? Yep. So maybe it got associated, but not anything strong. So then now we're bringing up this new new issue or new challenge, I guess is the word I want, where I need to continue to review that at some frequency, right? I need to look back and actually say, is there something more? So can you speak to a little bit, about like that struggle? You have more experience in this space, obviously, than I do. So what does that struggle look like, and what is the answer to that?
It's a struggle. I mean, it's a real challenge, and there's great potential for benefit in being able to do that, but there's obviously also a huge hurdle to do it. Mm-hmm. Who's got the time? Mm-hmm. How are we going to report back this information to the patient? There's sort of liability issues about this kind of thing. The burden of the challenge of communicating with the clinician. All of those things are outside of the need to be able to find this information altogether. And so there's a lot in the ecosystem that needs to be organized, but it's starting to become a topic of conversation. Many of the labs that we deliver Mastermind to have an approach that they're either developing or have already instituted for periodic re-review of the variants. There's two ways to do it. One is just a staged, like every 6 months you look back. Another one is every time you newly encounter the variant, if it's been longer than a certain period, then you would go back. But it takes extra time And it's sort of a situation where most labs aren't doing it now because it's challenging, but it's going to provide benefit to the patient. And so those two things are at odds. And that's just like I said before, that's a situation ripe for automation.
Yep.
And so Mastermind, as I mentioned before, being updated every week and having all this data in a database is a perfect capability to automate those look back needs. And so we have that in the form of Mastermind alerts. And as I said, we're starting to see an uptick in our particular larger reference labs using Mastermind alerts for that purpose. But that's a scenario where, as I said before, computational techniques can do the heavy lifting very sensitively, and then leave it to the human to review that information for specificity and accuracy, which is how Mastermind alerts works.
Yeah, I think that's a conversation that, at least within my previous life, right, with Fabric, we were having with every single hospital system.
Yeah.
What does that frequency look like? How do I report that back? I want notifications. I want to do it actively versus that notification being kind of more passively, right? Something new comes out, you tell me, you know. And so I think everybody's going through those debates right now, and so it's interesting to see how it develops. I think the next thing I was going to dig a little bit more into is specifically, especially with the paper with structural variants. I'd say that's probably one of the biggest challenges, and it's something, obviously the recommendations have come out for classifying CNVs, you know, not too terribly long ago in the world of time and space. So, you know, what is, so can you speak to a little bit about, you know, like CNVs and fusions, right? So those are very different, but very difficult problems to solve. And how does one review literature of that?
Yeah, I'll talk to fusions first because it's a little bit of a cleaner scenario. It's easy to understand, I think. Most of the time, a rearrangement or a fusion takes the guts of one gene and links it with the legs of another, and they get together and they cause havoc. And so those are most frequently described as a gene-gene event. So those aren't that hard to find, although increasingly there's a need to see the breakpoints and to understand really, you know, at a detailed level what's going on in that individual scenario. But it's also the case that as a graduate student, in fact, I discovered a fusion in the context of a cutaneous lymphoma. As soon as I looked at the data, I was like, this is real. This is a jackpot, because they're just not that common. It's a very complicated mechanism of causing disease. And so if the data says there's one of these, there's a high selection bias in favor of it being real. And the same is true on the clinical side. If you see, you know, an NPM1 translocation, and the patient has the appropriate disease, it's probably real and causative. So I would liken the identification of fusion genes to what we talked about before as the cancer challenge, which is to say, okay, now what? It's easy enough to find, but what else do you want to know? And I guess I should caution that just because I think it's easy to find with our computational capability doesn't mean that it's in all these other databases. So actually, in fact, that fusion event that I found is not found in other databases, despite it being published and published on, you know, subsequently. So it's a real causative fusion event. It's just not forthcoming in some of these other organized databases that are manual and out of date, or, you know, or lag behind for whatever reason, where it is in Mastermind. So that's the fusion component. But CNVs is a totally different animal. And I should emphasize, fusions cause disease at a real but somewhat lower level than, say, mutations, when you amortize that across all different diseases. CNVs cause disease in a much larger percentage of cases across all different disease types. So we're talking, depending on the disease, you know, 5 or 30% of cases, pardon me, are caused by CNVs. So the challenge with CNVs is the nomenclature is hard. There's a lot of colloquialisms. We're talking about named entity recognition in the context of AI. Colloquialisms, you just say, oh, this gene was deleted. You often will not use HGVS even though it's appropriate. Instead, you'll say exon 8 was, you know, cut out of this gene or whatever. So that's a challenge to find. And it gets even worse when people start to use chromosomal coordinates, which you know, you can describe in multiple different ways and makes it hard to index with a computational approach. And then also the bigger deletions like gross aneuploidies or cytobands that are amplified or deleted. So there's that heterogeneity in the description of the CNVs, but what I really want to focus on is how weird CNVs are in that you don't need to have the exact change.
Right.
In fact, you're far less likely to have the exact change as what you see in your patient. So there's interdependence in the data that we index that the user will want to see. If you've got a very similar CNV, I search for this region and there's a paper that talks about it like this, or there's a paper that talks about it like this, but it causes disease, or one of those larger cytobands that has a similar phenotypic presentation of my patient. There's a challenge in first finding all those and then presenting them in a meaningful way. And so the paper touches on our capability. It actually was published before that capability went live, but now that CNVs is live, I feel like our users are finding great value in it. And we're only getting better with the way that we visualize the data and the sophistication with which we index it, and the value that we can provide for those cases where CNV is probably positive.
Yeah, that's always been a challenge. Like, what is an overlap, right? Like, it's 90% overlap. Are you talking evidence with CNV? Were you going CNV to evidence? Like, what 90%? It seems like protein domains would matter. So yeah, I've been seeing that as a huge challenge in the space in order to organize that type of data and search broadly enough that you actually get the information. Yeah, I think we've got some questions coming in. So what I was going to do is just jump to the last closing questions that you and I actually put together. One of my big ones in all of this is, and what we were hearing, at least at Fabric, is time. A lot of this comes down to, time, right? So how long does it take variant scientists to get through and actually analyze all of these papers and whatnot to get through, because they want to return as many results to patients as they can, right? That's going to help them diagnose and help these patients as quickly as possible. So have you analyzed a lot of like the time savings that comes from these types of AI approaches, especially around like prioritization, right?
Yeah. So we've done a couple of studies, and so two of our users, about the time savings that it takes. I'll refer you to the paper where I think I described some of the time savings. We've also written a grant in the service of curating the genome, where we say it's only possible when you expedite things or semi-automate those things, as I talked about with our computational approach followed by expert curation. And that actually allows me to say that the time is actually, you can break it out into components. The database organization, that's been automated. So that's just straightforward bioinformatic. If you see this in your patient, assay results, go to this database and that database for population frequency and predictive data, that happens automatically and is highly quantitative, and so can even be figured into, say, an ACMG framework. The real challenge, though, starts when you talk about the published evidence, which is, again, where the crux of the evidence comes from for these interpretations. Right. So that itself can be broken out into two major phases. The first is searching, finding, organizing. And then, so that's what I call the muscular time component, and that takes the longest time. And then there's the cerebral component, where once you've found everything, you've prioritized it, you know where to look for functional or clinical stuff, this is not a good paper, this is a good paper, then you just look at the evidence and figure that evidence in the context of the ACMG framework. Yes, this is a cohort of patients who have a higher allele frequency for this variant than in the controls, or this is a family where there's segregation of disease, or this is a case where they've actually followed on with functional studies. Once you have all those references, highly sensitively pre-organized, pre-annotated, the cerebral part, especially if the data is presented in a meaningful way to the user, the cerebral part then becomes the only time barrier. And I've sort of, in my own studies, fractionated that muscular searching component. That's the majority of the time that's spent. And that's what Mastermind and Genomenon writ large has largely automated. So we've shrunk that time component down now, so it's a highly cerebral exercise. So I joke with my curation team members that they're getting smarter because they're exercising their brain, but they're getting their body's getting deconditioned because they're not doing the muscular component so much because it's done for them.
Well, yeah, and it also means, the people, those people that are highly specialized that are being trained for all of these reasons
Oh,
are
yeah.
using their time in the right way,
Yeah,
right? They're actually
exactly.
able to look through more papers and aren't just, professional Google searchers.
So that's a great point. I mean, we are variant scientists on my team.
Mm-hmm. Yep.
And there is, I can actually sort of round out our part of this discussion by saying that I don't expect AI to take over the human component ever, particularly in clinical medicine, particularly with the complexity attendant with genetic and genomic information. It's the application of AI, as I've just described it, is all about the muscular component, sensitivity, organizing, annotating content, delivering the information in the most appropriate way to the user, but it will always, I think, rely on the human user to approve of that evidence if they're producing, say, a curated database, or otherwise validating that curation from the other user. And so the variant scientist position is not going away. In fact, as you alluded, it's only getting more needful. We need more of these people. There's just not enough of those folks appropriately trained to people to work for us.
Yeah, because we're moving people from, a single SNP assay, obviously, thing of the past, but we've moved to panels, the majority of sequencing, right? As we go from panels, we're going to get a whole genome. I mean, those people are going to become more important, which actually brings us all the way back to the beginning of it not being a black box. Like I've heard it said around, I love the words around Genomenon, show your work. Which is another way of saying that, right? Like it's presenting that information back in such a way as that a human, that variant scientist, that trained professional can go through and really analyze that data.
Absolutely.
So I think with that, we'll turn it over to Garrett. I believe you're going to jump back on and ask some of the questions that have been coming from the audience.
I am, yes. Mark and Brittany, this has been a really great conversation. We do have some questions that have come in from the audience that we'd like to get to. Everyone who's watching, if we don't get to your question, a member of our team will get back to you. So Mark and Brittany, a viewer wants to know, how do you classify variants from damaging to pathogenic? We carry a lot of damaging variants, but only a few could be pathogenic.
Yeah, great question. I think one of my latter answers talked about the ACMG and AMP framework. And so if you're not familiar, it's a very structured, it's very complicated, but it's a very structured analytical approach to reviewing and adjudicating the evidence. And so the viewer points out a good case is we have these neat, tidy categories of benign, likely benign, uncertain, which is a big, middle ground, likely pathogenic and pathogenic. And biology just doesn't work that way. We need it to, from a laboratorian perspective, we need to have that reproducibility. We can't say it's kind of this way and we think we might do this, because you actually have to take action. You have to make a diagnosis or not. You have to treat the patient or not, et cetera, et cetera. And so in a way, we're sort of forcing these categories onto what is more of a spectrum. And so in particular, when we talk about variants of uncertain significance, there's a spectrum of variants of uncertain significance. There's some that have a modest amount of evidence, but insufficient to go over the threshold to be deemed likely pathogenic by ACMG. And then there's others where there's a lot of evidence, but it's still sub-threshold. And so I think the viewer's question is sort of touching on that idea that really there's a spectrum, and the ACMG framework is really intended to put the decision in the hands of the user in context-dependent terms. There's other clinical parameters around this patient and their presentation. There's other circumstances that will guide the variant analyst and or the clinician into making the most appropriate determination. And sometimes that can mean, yeah, it's technically a variant of uncertain significance, but the clinical circumstance would dictate that my action as a clinician will be to treat because of these circumstances. The clemency of the treatment, its effectiveness, the fact that you have other, family members who are affected who have the same mutation, et cetera, et cetera. So Brittany, I don't know if you want to build on that.
I mean, just that, I think you really said it with it is a spectrum, right? They're trying to give you some type of guidelines. And I think one of the great things about using some of these, using this type of approach that we're doing where we present you back evidence in a very organized manner is that you can actually normalize across different people.
Yep.
Right?
Yep.
Because what you don't want to see is that, this person over here, even in different labs, this person over here uses that framework and says, this is likely benign. This isn't actually anything we're concerned about. And then somebody else uses that information because it's just a framework, but there's still so much variability in it, that somebody over here says somehow this is likely pathogenic. If we can
showing your work,
all information back. Yeah. The more information that those people can consider, because if this person was missing half the information, they may call that likely benign, whereas this person had all that information and says, no, there's really a lot more.
Yep. Good.
Very good. So our next question is, hi, could you explain GLP in detail?
Hi. Oh boy. Well, I mean, GLP is a living thing. It's been accumulating facets and capabilities and improvements since Genomenon's founding, which is actually 7 years ago yesterday, for a little bit of history.
Happy birthday!
Yeah, let me do my best to do it succinctly, but in sufficient detail where you get more information than I provided before. So I alluded to the way that we find genes with all of the synonyms and aliases and nicknames and previous versions, et cetera, et cetera. So that is the sensitivity part. There's also the need, once you've found a gene in one of these references, to know if it is in fact a gene or it's just it's a true technical match, but it's a false sort of biological match. An example that I like to give, a better example than the one I usually give, is SDS is a gene, but it's also a detergent that's used all over the place in blots and dishwashing and that sort of thing. So GLP not only recognizes these things, but is context aware to say, yeah, but it's not the gene that's being referred to here. But there's still a healthy balance between sensitivity and specificity, and so we take great pains to strike the right balance of being sure that we can throw away stuff, but not so much that you lose out and throw some babies out with the bathwater. So that's the gene level. But just like I said before, like you have a gene in a paper, you've also got variants in the paper. There's the need to be sure that they go together. And so GLP is, as I mentioned, bioinformatically literate. It knows what the nucleotide is at this reference position in this transcript for this gene. And so if you have these three genes had these three mutations, and the challenge for NLP or GLP is to connect that gene to that variant, it does so fairly sophisticatedly using the genome sequencing information, using context clues, as well as using the data that exists even outside of that paper. Is it more likely to be this variant because that variant was seen before? And then the last thing I'll say is, and really the heart and soul of GLP, is how sophisticatedly we can index the variants. So an RSID is typically a polymorphism. That's an easy thing to find. It's just RS number. It gets much more complicated when you talk about cDNA variants. There's multiple ways to describe a cDNA variant, complicated by transcripts and legacies like signal peptides and that sort of thing. But then there's also the protein level descriptions. And I mentioned V600E before in some of my answers. Simple, right? Well, there's really about 120 different ways, like completely distinct ways that variant has been described in the literature. And GLP is aware of all of those and is able to accurately identify, yeah, this is BRAF V600E. It belongs in this result set when the user types in whatever one of those 120 ways they type in for their search. So hopefully that's enough of a flavor of GLP, but then it also has diseases, phenotypes, drugs, categorical keywords, et cetera, et cetera. So I just gave you the sort of tip of that iceberg.
Okay, so in our last 6 minutes here, we probably have time for one more question. So do you foresee Mastermind as replacing ClinVar or some of the public databases?
I don't. And in fact, I've had conversations with folks at high levels in ClinVar, and we're not. It's a different purpose. Mastermind does not receive patient information. We don't receive variants from users. That's what ClinVar does. And so but moreover, ClinVar doesn't curate. They don't put an emphasis on organizing the evidence, being very sensitive about those evidence citations, and they're a repository of information. They don't supervise what the users submit. So I see them not as competitive at all, and not one is better than the other. They're just for a different purpose. And actually, what I'd like to see is more of an integration of the benefit of ClinVar from the live flesh and blood clinically submitted data, as well as the empirical evidence-based, archival data from the literature that only gets more and more proliferative. I mean, we're focused on this, and we're doing it really well. And ClinVar is focused on doing this and doing it capably. And so I see a great benefit to merging those capabilities for the benefit of the community. Brittany, I don't know if you want to speak to that too. Do you want to take?
No, I think the exact same thing. No, I would say the exact same thing. I think there's always going to be utility in that, people submitting, and also with that, you get to really just at a glance see who that was, right? So you get to at a glance see that this information came from lab 1 or lab 2, and I trust that lab. They're submitting criteria answers. There's different types of information that are available there versus, the breadth, the sensitivity of information that Mastermind can show, because that's from all literature, including, supplemental information.
Yep.
That may not be something that anyone's ever going to deposit in ClinVar, but hopefully we can build ClinVar.
You're great.
Thank you both. Do you guys have any closing thoughts or?
Oh, again, Brittany, welcome aboard. It's really great to have you. It's funny. I feel like we're old friends. We've only had 2 hours worth of conversation, and this is one of them. But it's good to have you on board.
Well, no, my one closing was you keep saying this. Genomenon's mission is to curate the entire genome. So get on that. When's that going to happen?
Jeez, already cracking the whip. Well, I told you we're taking pains. We're starting out with a prioritized approach.
Yep.
And I think we'll surprise people with how quickly we do it.
Yep. Yeah. And that speaks to the more people that throw more information into publications, which is great, more scientific knowledge, our work will continue to be, because we'll have to index even more. So.
Yeah.
Cool. Well, to everyone who is on this webinar, thank you for tuning in. And again, this webinar is recorded, and we'll send that to you soon. Mark and Brittany, thank you so much for taking your time to offer your expertise. If you have any questions, feel free to send us an email at hello@genomenon.com. And if you haven't signed up for Mastermind yet, we have a bit.ly link on the slide that's up where you can create your free account and start with a trial of Mastermind Professional Edition. And we'll send you all this information in an email as well. At the conclusion of this webinar, you'll receive a short post-webinar questionnaire. We'd love to hear what you thought of today's event. So with that, I think we're wrapped up. Thanks so much.
Yep. Thank you.
Bye.
Bye.